using an integrative taxonomic approach. Over to you, Shilova.
Thank you. Hello, everyone. So as Lucanio has introduced me, I'll be presenting on the Cape Galaxias, where we're using an integrative taxonomic approach to delineate the different species. So the Cape Galaxias is a freshwater endemic fish that is endemic to the Cape Fold ecoregion. This species have been considered to be the only representative of the family Galaxidae in Southern Africa, and it has been thought to be widely distributed all the way from the Olifantsdorring to the Gamtoos River system in the Eastern Cape. However, the introduction of molecular studies have revealed that there are at least 15 distinct lineages within this single widely distributed species. But it has been since the first publication by Waters and Cambry, which was in 1997, that it has been known that there is undocumented diversity within the Cape Galaxias, but there has not been like morphological diagnosis that have been provided to link up the different lineages. So the aim of our study was to use an integrative taxonomic approach, which incorporated the molecular and morphological data to determine the taxonomic distinctiveness of the different lineages that were identified within the Cape Galaxias, with the main objective to use this data to provide formal descriptions of new species. So just to give like brief molecular results, so we recovered 16 different lineages within the Cape Galaxias, and these lineages are all distributed within the Cape Fold ecoregion. So from these lineages, we selected 3 morphologically variable lineages that we could easily like separate them like amongst the rest of the 16 lineages. And these 3 lineages that we assessed was the Stiembras, Rifisonrand, and the Hoku. And we compared the morphology with the true zebratus. So we also selected the Stiembras, Rifisonrand, and Hoku because each of them are found within a single river system. So it was easy to delineate. And also the fact that none of them were coexisting with the true zebratus. So thirdly, we selected Rifisonrand and Hoku because they were classified as highly threatened according to the IUCN Red List assessment that was done in 2017. So the morphological results show that when looking at meristics, with the exclusion of Hoku, these lineages cluster together, so they're not really separated from each other. But then when looking at morphometrics, these 3 lineages can be separated from each other as well as from the true Galaxias zebratus. And the characteristics that were outlined as the characteristics that are mainly informative for this separation was the eye diameter, post-orbital head length, head width, pre-dorsal fin length, and the length of the cauda peduncle. And then we compared these 3 lineages with the true zebratus, of course, while looking at the morphological variation and the distribution. And we saw that Galaxias zebratus was recorded from multiple river system, and it has distinct vertical bars along the flank and rounded to subtruncal caudal fin, whereas Stiembras was recorded from a single river system, and it has brown body pigmentation with densely scattered spots when compared to the true zebratus, and a more truncate fin. And then thirdly was the Hoku, which was also recorded from a single river system, of course, and it has a body that is covered in brown spots of varying sizes and a more indented or imaginate caudal fin. Lastly was the Galaxias Rifisonrand, which was also collected from a single river system. It has fading irregular vertical patterns when compared to the distinctive ones in the true zebratus, and a more truncate caudal fin when compared to the Hoku that is more indented. So why is it important for us to provide these formal descriptions for the different lineages? Firstly, these lineages are a biodiversity heritage, and this is especially important in the Cape Fold ecoregion because this place is known as a hotspot for endemism. Secondly, it's for biodiversity conservation in that accurate species delimitation help identify species that are at risk and guide IUCN Red List assessment. Lastly, it's for environmental management in that native species serve as bioindicators for river health and ecosystem integrity, and that accurate biodiversity data improves spatial planning. So all in all, we provide these formal descriptions because we cannot protect what we don't know. Here are my acknowledgements. Thank you.
Thank you so much, Shilova, for a lovely talk. I'll take one question because of time. Okay.
Hi, congrats. Very good, your presentation. Maybe because I don't know the area, I'm wondering if this amount of lineage is because the river, they are not connected or something like that. But can you explain more about this, please?
So let me just go back. It will be easier to explain with this diagram. So these rivers, so different lineages, most of them are found within a single river system. So this river system don't connect with each other. But we have the likes of the red one, which is Galaxias dubius, which is found within multiple river systems. This one, there's been studies by Albert. I'm sure if you want to hear more about why it's distributed like this, he can explain very well. But then in his paper, he looked at area exposure as whole. He wanted to know why it has this special, like this wide distribution. So he looked at area exposure as in like, are they able to breathe outside or when they're immersed? Is it immersed? Yes. So he found that they can actually survive outside for like 36 hours max. Yeah.
Thank you.
Thank you so much, Shiluva. Please, round of applause for Shiluva. Please allow me to introduce our next speaker, Esihle. She will talk about the phylogeography and genetic differentiation of the Bao stripe across its range in Southern Africa.
Good afternoon, everyone. I'm presenting on the phylogeography and genetic differentiation of the Bao stripe minnow. So the genus Enteromius is endemic to Africa. In Southern Africa alone, it is represented by 43 valid species. And there are still ongoing studies that aim to describe new species and to revalidate species. And there are some recent studies that have revalidated species like the Enteromius crocodilensis, and also describe new species like the Enteromius nichen. So there is evidence that many of these species within this genus harbor undocumented diversity. For example, the Enteromius viviparus. So the Enteromius viviparus has a wide distribution from the Ruvumo River system in the northern Mozambique down to the Uvungu River system in KZN, South Africa. With species with this wide distribution, such as this, previous studies show that they tend to harbor high diversity. So the question is this the similar pattern with Enteromius viviparus as well? So this study builds on previous studies, and it is to document the extent of undocumented diversity within the Bao stripe minnow across its entire distribution range. So the objectives is to incorporate samples from river systems representing the full distribution range and identify unique lineages within the species, and to determine the geographical distribution range. So the results show that the true viviparus in yellow clustered with the major clade with other Goldie species and related taxa, and it is distributed from the Pongolo River system down to the Uvungu River. And the second clade, which again divides into two other lineages, which is the slender bow stripe and the northern lineage. They all actually had a genetic distance of 4.4 to 9.1%. But interestingly, these two new lineages are distantly related from the true viviparus, even though morphologically for a long time they were said to be the same species. And the slender bow stripe is found from the Inkomati, Limpopo, up to the Zambezi River system, and the northern lineage is found in Polombe River and also the Montepies River system. So This study shows that the DNA barcoding remains a powerful first-line tool. It helps uncover hidden diversity and provides clarity on identifying specimens across distribution ranges. And the study is evidence that these species have much narrower distribution ranges as said to be. And this is essential to improve the accuracy of the extinction risk assessment. So further morphological studies are done to confirm whether the genetically distinct groups represent separate evolutionary lineages, and also if to support the description or the revalidation of new species. Thank you. These are my acknowledgements and
Thank you so much, Sister, for an interesting presentation. Any questions from the floor? Online? Come on, people. My guy. That's it.
Good afternoon, everyone. I'm the odd one out here. I'm a terrestrial biologist, so I might add some things that you guys may know. I wanted to ask, even relevant to the previous speaker, about are there like real color morph differences between these different species? Because I don't know, I looked at the different descriptions of variations of brown kind of thing, and I don't know if they occur in these different places, do you have like environmental factors that influence how they appear? Do they look the same? How do you really distinguish when you see them and say, okay, look, these are like different subclasses, or is it like it can change in an individual's lifespan or whatever? Thanks.
One of the motivations of the study is because it was recently divided into two morphological groups because of there's some morphological characters that seem to differ between them. Even when you look at Paul Skelton's 2024 book, you can actually see the slender bow stripe is different from the true viviparous or what he describes as true viviparous. And also, yes, environmental factors affect the fish morphology so much because in cases where you find a species in turbid waters, so you'd find the eyes are bigger than the species that you find in much more clear water, which it has small eyes because of trying to find food. So there are those morphological characters that you can look at that you explain, because morphology alone, then you explain why this changes occurred in this individual. And there are some species that you find that because they are very specific on their environment, even though they are separated, their distribution separate, but you find that they still have same morphological characters. So yeah.
Thank you so much, Sister. A round of applause for it. This next speaker needs no introduction. Luleko Nzuma, Msholozi. Ladies and gentlemen, please welcome Kululeko. He will give a speak talk about genetic diversity and hybridization between native and introduced Oreo from a species in Southern Africa. Take it away, sir.
Thank you for the riveting introduction, Lukanya. So today I'll be telling you about my PhD project that I started this year titled Genetic Diversity and Hybridization between Native and Introduced Oreochromis Species in Southern Africa. So to give a brief background, aquaculture is a booming industry in many parts of the world, but it is an area of potential development in South Africa. So the government has been engaging with the aquaculture community on the possible use of Oreochromis niloticus, and aquaculture permits have been issued in Gauteng, Northwest, the Western Cape, as well as the Eastern Cape. But the development and the permitting for the industry has been highly debated in the Limpopo, Mpumalanga, as well as KwaZulu-Natal. So, Oreochromis niloticus is considered a conflict species, meaning that it can confer like great economic benefits, but it can also cause many negative ecological impacts. For instance, Oreochromis mozambicus has been listed as vulnerable on the IUCN Red List here in South Africa due to its hybridization with Oreochromis niloticus. And as well in Lake Kariba, most of the populations of Oreochromis mortimeri have been extirpated due to the introduction of Oreochromis niloticus, but some populations still persist in the Middle Zambezi. Just to give a broad overview of the natural distribution of Mozambicus. So it's distributed from the lower Zambezi system right down to the Bushman system in South Africa. And for Niloticus on the right, its natural distribution is in the Nile Basin as well as the Rift Valley lakes and some rivers in West Africa. So the aim of my project is to map the distribution of pure populations of Oreochromis mozambicus in South Africa, and as well as Oreochromis mortimeri in the Zambezi Basin to guide sustainable aquaculture development in the region. Another aim is to determine whether Oreochromis mozambicus is a species complex or is a single species across its broad distribution range in South Africa. And my objectives include to test the potential application of low coverage whole genome sequencing on a regional scale. Another objective is the development and optimization of a tool that will be used for rapid assessment of the genetic integrity of other Oreochromis species in Southern Africa. So how will we do this? So sampling and morphological IDs have already been done in the Popo, Mpumalanga, and KZN by the NRF-SAIAB team in collaboration with the SANBI as well as the DFFE. And now we'll be expanding the sampling into the Eastern Cape. But there's difficulty in differentiating hybrids, basically using the morphology. So we'll be using genomics. And in genomics, I'll be using low coverage whole genome sequencing as well as Oxford nanopore sequencing devices. And this will be followed by data analysis to determine genomic variation as well as population differentiation. Some of the results that we might expect is the fast structure bar plot, where multi-carded bars will indicate hybridization, as well as the principal coordinate analysis, where individuals that fall outside of the cluster will be indicative of hybrids. So my expected outcomes is to provide information that will aid implementing agencies and issuing permits, as well as address the question of whether Oreochromis mozambicus is a single species or a species complex, as well as develop a tool that will allow for rapid genetic assessment, as well as develop a conservation plan for Oreochromis mortimeri in the Middle Zambezi, if we do find. So here are my acknowledgements, and I would like to thank NRF, NRF-SAIAB, Diplomix, and as well as SANBI, as well as DFFE for providing funding, as well as the other departmental. Thank
you so much, Pulilego, for a great talk. Do you have any questions from the floor?
Sure.
Actually, it's not a question. I want to add something to the project. Also in Oman, since I used to work on freshwater fishes of Oman, there are lots of Oreochromis mozambicus in morphology, but by genetic they are Oreochromis niloticus. So it is maybe it could be a hybridization in that country, which has a vast distribution there. So I think it's a board issue in this case.
Yes, thank you. Thank you for the comment.
For my question.
Well,
I wanted to ask.
Sorry, you still.
I wanted to ask. Worst case scenario, you find out this other species in Mozambique is invaded the system. What rehabilitation options are there? What do you do with the river system that has this species dominating that shouldn't be there? After you found that out, like, what can you do to reverse that?
Well, it's actually Niloticus that is invasive. So this is a difficult one. It could be we could like look into like actually like fishing fishing them out of a particular system. But because here we're looking at, aquaculture development, if we like find them in, let's say, already in an area or particular river system where it's present, or we also find hybrids there, it's that area where we'll be actually giving permits for like farmers to actually do the aquaculture work. And in areas where we only find Oreochromis mozambicus, we do our best to provide like a conservation plan for that particular river system. And the implementing agencies may then prevent the issuing of permits in those regions.
A round of applause for Kulego. Thank you. Please, I would like to introduce our next speaker, Nobuhle Mpanza. She will talk to us about unpacking the functional roles of mussels in coastal habitats. Floor is yours.
Good afternoon, everyone. Thank you for the intro. So today I'm going to be giving a presentation on my collaborative work with the University of Plymouth, which I got the opportunity to visit earlier this year. Just a brief background. We live in the Anthropocene age where we're seeing our natural coastal environments being transformed into those man-made artificial infrastructures which lack important microhabitats. And these microhabitats are important because they're complex in structure and they favor a number of species within the intertidal zone and coastal environments. Losing those important microhabitats means we're losing the species and the overall functionality of these ecosystems. So what are scientists trying to do? They're using ecological engineering to kind of bring back the complexity within the artificial infrastructures in urbanized coastal environments. And my work focused on mussels, but how do mussels fit in within the scope of ecological engineering? So mussels are known as ecosystem engineer species, which means they are habitat providers. But what they use for in ecological engineering is they seeded onto these complex infrastructures where they kind of bring in an additional substrates for other species to come and attach, as well as also, they're also important in terms of purifying the water's quality within the systems that they found in. So my work took place at the Queen Anne's Marina close to the University of Plymouth, where I looked at two types of infrastructure, including pontoons as well as wave screens, which are most common structures across the globe within urbanized coastal environments. I looked at two specific things. Firstly, I wanted to look at the overall density of mussels within pontoons and wave screens. And I also wanted to look at the size distribution of these mussels, as well as the biodiversity of species that are associated with these different environments. And secondly, I wanted to look at clearance rates in terms of water quality, looking at how mussels with epibionts filter water compared to mussels without epibionts. So firstly, when it comes to the biodiversity parts, the mussel parts of my study, I used quadrats to deploy on the pontoons as well as on the wave screens. And then I assessed the bed density of the mussels as well as the epibiont diversity on the actual mussels as well as on the surrounding environment. But I also looked at the size distribution of the mussels that we're finding between these different type of habitats, as well as the prevalence of the epibionts, whether they're covered by the epibionts or they're not covered, as well as how much of the mussels that I've collected are actually covered by epibionts. And then this is just basic results that I have so far, because I just finished this research in August. When you're looking at pontoons, there's not a lot of biodiversity, but when you're looking at the wave screens, there's more biodiversity compared to the waves, to the pontoons. And it also comes with a lot of epibiont diversity, as well as mussel size and the density of the mussels within those size. And then in terms of the size of the mussels, when we're looking at wave screens, we found, if you're looking at the orange on my federal rights, the orange graph, We found that wave screens are supporting large mussels that are in dense bed densities, as well as whereas the pontoons are doing the opposite. When it comes to the epibiome diversity, the mussels that were found in the wave screens were also supporting high densities in terms of epibiome diversity, as well as the diversity overall. And then the second section, of course, was the clearance rates, where I was looking at water quality in terms of how much the mussels are eating. And then I collected my mussels, filtered the water to ensure there's no food remaining in the water that I'm going to use for my experiments. And then I had to acclimatize the mussels for 24 hours before I can conduct my experiments. And these are different treatments that I had. If you're looking at the top ones in green, those are all live mussels with different densities of with different percentage cover of epibionts, and the epibiont that I chose specifically for this study was barnacles, because they're also filter feeding species. Then if you're looking at orange, those are all dead mussels. It's just a replica of the ones in green, except the ones in orange are dead, just a replica of the other ones. And then I also had my controls. So firstly, for my feed, I use commercially obtained isocrasis algae, which I had to prepare and feed into my chambers. So each chamber had individual mussels of the different treatments that I just stated. And also I had to feed each of them at different time intervals, and then extracts for 2 hours. I had to conduct the experiment, and then I had to extract 20 moles from each of the chambers and then into the sampling ports, and all that went into the culture counter machine, which basically just reads how much is there at time 0, how much is there at time 15 until 2 hours. So you're just looking at how much, if I'm putting a mussel in a chamber, how much is it eating over time for the duration of the 2 hours, and how much are the epibionts that they're carrying, are they eating as well? So far, these are the basic results that I have. All the ones here are the live mussels, which I've highlighted in green, and you can clearly see that if a mussel is alive, because of the filtering ability that they have, they eat more of the algae that has been provided. But what you're seeing also, this one is when there's a high load of epibionts. The feeding does not stop, but there's just a reduction in the feeding rates of the mussels. And all the dead ones, they're also functional as not as filter feeders, but as secondary habitats for other species such as the barnacles, which are also filtering. So you can see some type of filtering still happening within the treatment where the mussels are dead. And of course, there's no feeding on the controls. So firstly, what does this mean? The wave screens had high densities of mussel beds with large mussels supporting high diversity of epibionts as well as the surrounding species. So a number of things are important to note. It's the zonation and the hydrodynamic factors that are acting between these two different habitats, but also the structural complexity was very important. So when it comes to ecological engineering, structural complexity is the main factor which people try to look into and try to increase. So it was also important to see when you're seeing the wave screens, which are more complex compared to the pontoons, that's where you're seeing the success of mussel growth, the success of other species colonizing those areas, as well as another thing that was important is the management plans that are in place within harbors and marinas also play into factor in terms of the biodiversity that exists in these environments. For instance, on the pontoons, we had the issue of the clearing of the mussels. So there's regular clearing of the mussels, which makes sense why we're finding less density, less mussel bed densities, as well as small sizes, which contributed to the low diversity that we're seeing within the sites. So one important thing when it comes to the clearance rates was the fact that whether a mussel is alive or dead, it's still a functional species. So if it's alive, it's going to function as a water clearing species within that system by filtering, taking in all the impurities in the water, eating. Whereas if it's also dead, it's also functioning for epibionts that are existing within the system. So it's providing habitats for other species, but it's also one knock-on effect is the fact that the species that are actually attaching on the mussels, they're also acting as filters themselves. So the process of clearing the water within this man-made urban environment still continues beyond whether mussels are alive or dead. So this was a collaborative efforts that I got the opportunity to have with the University of Clement, and it was funded through Leviathan Grant, which I'd like to thank, as well as all the Andrews, which are the technicians responsible for my training when I was in Clement. Thank you.
Thank you so much, Dr. Chuk. Any questions from the floor? Now, look, we have people online. Just need a mic here.
Thanks, Nobu, for your presentation. I think you said, if I'm not mistaken, the feeding rate was affected for those species that were heavily carrying the epibiotes.
Yeah.
So does that also mean the growth rate is affected by default? Do you know that?
I looked at the size of the mussels. So not the growth of the mussels is not affected. It's just the filtration rates that's reduced, because we're finding way higher or larger sizes of mussels within the pontoons, within the wave screens, even though they were fully covered by barnacles.
Oh, okay.
So it's not affecting the size at all, just the filtration.
So my next question I was going to ask about the settlement. Did you notice the size range? Were they selective, the barnacles of which size are they settling on?
Yeah, I think one of the graphs that I displayed, maybe I didn't talk much about it, but if a mussel is larger, there's more surface area for species to come and use them as a habitat, a secondary habitat. So larger mussels, more room for other species to come in. So the sizes within the wave screens were much larger, which also contributed to the high diversity of species that you were seeing there.
Last question.
Thanks. That was a really cool talk. I wanted to ask you, in line with the clearance experiment, if you had to do a similar experiment in the field and not just look at clearing of algae, but clearing of sediments and maybe heavy metals and plastics and things like that, would you expect to have the same effect? Because I know oysters can clean water. Do mussels have the same kind of property?
Mussels also are cleaners of water, but I'm not too sure when it comes to metals. But I know there's a lot of work on the clearance rates, filtration rates of mussels. So they're very good at cleaning our environmental areas, especially in urban areas, which is why maybe they kind of used into ecological engineering interventions.
Thank you.
Close for question. Our next presentation is a pre-recorded talk by Chapulani Ndaba. He will talk to us about the influence of diatoms, water quality, and early-stage food webs by eco-engineered designs.
Good day, everyone. Due to time, I'll be only focusing on the microphyto benthic component of naturally made eco-engineered designs. So natural coastal systems provide a wide range of provisioning services such as primary productivity that sustain human livelihoods. However, increasing anthropogenic pressures such as urbanization and industrialization along coastal systems have resulted in the replacement of natural rocky shores to less complex habitats such as seawalls. And this is known to negatively affect the initial colonization and successional patterns of coastal species, subsequently impacting ecosystem functionality and resilience. So changes in habitat structure are particularly more pronounced at the base of the food webs in lower trophic groups such as biofilms, which are ubiquitous masses of microbial communities that are dominated by diatom species in coastal environments. So diatoms are essential as they account between 20 to 40% of the global primary productivity and act as a middleman in providing energy sources to higher trophic groups, therefore dictating the structure and dynamics of coastal species. So numerous studies have indicated that diatoms are highly sensitive to environmental as well as structural changes, with comparison between artificial artificial versus natural environments, indicating reduced species composition and diversity, as well as an increase in the dominance of specific diatom groups on artificial substrates compared to natural, leading to functionally altered biofilms. So these changes have a negative cascading effect on coastal food webs as well as ecosystem processes. So in order to mitigate or minimize changes in habitat structure, ecological engineering aims to restore structural complexity on artificial substrates by incorporating biogenic elements such as bivalves and structural elements by mimicking natural habitat features such as rock pools and crevices. So several studies have indicated an increase in species diversity and abundance on these designs. And while most ecoengineering studies have mainly focused on conspicuous taxa such as microalgae, fish, and bivalves, microalgal assemblages such as diatoms have received less attention, with only two studies that have considered them. So the first study is by McManus 2018, and the second study is a study we published earlier this year. So this presents a knowledge gap given the foundational role of microalgae in coastal environments, and in also understanding how eco-engineered designs facilitate the early colonization, diversity, and functional traits of diatoms. So the present study aimed to assess diatom species as well as functional diversity on two designs across varying coastal environments, and hypothesized that the structural and biogenic complex treatments will have greater diatom species and functional diversity due to the provision of a more heterogeneous micro habitat, which will enhance overall biofilm development. So in this study, we had two designs. In each two-design sampling period with 3 treatments. So the first design is the Emisi design made from Cyperus textilis using indigenous artisanal practices. When in this design we had a control, which was the side-scraped rock, and heat treated. We had a flat, which is a structure without seeded, and we had a seeded treatment, which is a structure seeded with live penna mussels. The second design was the salt saver structures, which were made from natural wood fiber, similarly consisted of one control, a flat without mussels, and a seeded with mussels treatments. So we conducted monthly sampling at 4 sites, namely at St. Francis, at Port Elizabeth, Port Alfred, as well as Port Nocha, where 38 centimeter squared areas were scraped in order to collect the biofilm materials. So biofilm samples from month 1, 3, and 6 were processed for the study. So when reaching the lab, the samples were processed in hydrochloric acid as well as nitric acid, and diatoms were imaged using DIC microscopy, and diatom valves were counted. So species were identified up to genus level grouping. For statistical analysis, biodiversity metrics such as richness, Shannon's index, as well as evenness were calculated, and for functional matrix, the diatoms were initially grouped according to their morphological guild, their motility, as well as their life forms, where functional richness, functional evenness, as well as functional dispersion was calculated. For statistical analysis, 2 approaches were used. A Permanova as well as a mixed effects modeling approach was used. So just to show you some of the diatoms were identified, so I selected some of the beautiful micrographs I've taken, which include species such as Campylodiscus, Astrococcus, Agnatha species, as well as the Chromatophora species. So when we look at the diatom commune assemblages, we did find a significant effect of site and treatment in both designs. As seen in the figure here, there is groupings according to site in both designs, as well as separation between treatments in both designs with a bit of overlapping observed. When we look at species biodiversity metrics, we did find significant differences in species richness and Shannon diversity in the soil saver design, which were both higher in the flat and seeded treatment compared to the control. In the soil saver design, we only found that the species richness was significantly higher in the seeded treatment compared to the control. For evenness, we found that it was only significantly higher in the seeded treatment compared to the control for the immediate design. For functional matrix, we only found significant differences in functional richness as well as functional evenness in both designs, which was significantly higher in the seeded treatment in the immediate design as well as in the soil saver design. So in the study, site and treatment had a consistent effect that shaped diatom species composition. So the significant site effect can be alluded to the site specific conditions such as hydrodynamics, which including wave action, and environmental condition that could include pollution levels, which are known to influence diatom species colonization and successional patterns. The significant treatment effect in both designs can be related to the structural complexity in both the design by providing clear microcrevices and sheltered microenvironment seen in the immediate design and the coarse fibrous texture in the soil saver design, with both designs providing attachment sites for diatom genera to colonize at different scales. So when we look at a design scale, the mesostructure significantly enhanced species richness and diversity, suggesting that both structural and biogenic complexities increased available niches by promoting microhabitats and also possibly through nutrient enrichment from mussel deposits. So this was further observed with an increase in functional richness from the seeded treatment, which shows expanded niche differentiation and ecological roles that could aid in regulating services such as water purification as the diatom community develops over time in this design. In the soil saver design, it indicated a consistently higher species richness as well as functional evenness only in the seeded treatment, and this indicates that the biogenic facilitation was important in promoting diatom species and traits, similarly by nutrient enrichment from mussel deposits. So the significant functional evenness in the immediate design indicates that the diatom traits were evenly distributed, suggesting that multiple diatom traits are abundant, and this enhances resilience to change as the diatom community stabilizes and develops on this design. So in conclusion, the increase in diversity and functional diversity matrix was treatment-driven, demonstrating that both the immediate and the soil saver designs are suitable plant-based eco-engineered designs. However, the immediate design was more effective as it yielded more diverse and functionally richer and even diatom genera, while the soil saver design was less effective. So moving forward, eco-engineering studies should implement diatom species in their biodiversity assessments and also consider other factors such as water quality as well as hydrodynamic factors. I'd like to thank and appreciate the following institution for making this research possible, and I'd also like to thank the team for its assistance. Thank you.
Thank you so much, Shabulani. I hope you're still there. You didn't run away.
Yes, I'm still here. I'm still here.
Okay, thank you. Do you have any questions from the floor? Questions, please, online. Okay, thank you so much. Question there.
Nice presentation, Jabulani. I just wanted to ask, the prototype that you guys have created of immediate and soil saver, is there potential for upscaling? And if yes, what's the way forward from now on? I know you're submitting your PhD, but what's the way forward in case there is an opportunity for upscaling?
Yeah, thank you for the question. I hope you can hear me. I think based on what you've seen in terms of the results, there is a potential of upscaling, but I think there's other things we need to consider, such as reinforcing the designs in order for them to last longer. So there are potentials of upscaling based on the positive results we've seen in terms of the functionality aspect.
Thank you so much, Shabulani. I would like to introduce Violetu. She will talk to us about the biocultural and livelihood significance of Cyprus textiles in coastal South Africa. Over to you, Violetu.
Afternoon, everyone. So this is one of my PhD chapters. I'll just get straight to the introduction. So the non-timber forest products are known to sustain livelihoods worldwide. So these include a range of animals and plants that are harvested from the forest, savannas, and the natural ecosystems. So in South Africa, coastal communities, they value these NTPF for crafts, for cultural significance, and because they provide sources of income. Certain wetland species such as Jungus krausi, which is locally known as Ingema, and Cyperus magnetus, which is a desert sedge, and Cyperus textilis, locally known as imizi. They're prized for their physical properties that make them suitable for crafting these crafts. So the choice of plant material typically depends on the type of craft and the desired technique of weaving. And beyond their cultural and economic value, these plant-based materials, they have a potential of addressing sustainability issues. So one example is the imizi project that Jabulani just spoke about, where the multidisciplinary project where there's a co-creation of these woven mats that are made up of Cyperus textilis. So we're working closely with the local community known as the Keiskamma Trust. So these mats are installed in harbors and ports. The goal is to increase the surface complexity, enhance biodiversity, and ultimately rehabilitate urban coastal environments. So the sedge Cyperus textilis, which is locally known as imizi, and I will continue to refer it as imizi as I go through the talk. It is an indigenous sedge found in the southern parts of Africa. It is distributed from Western Cape to KwaZulu-Natal. It is a facultative wetland species that grows competitively in freshwater environments. It mostly found in riverbanks and streams and dams. So this plant traditionally has been used to weave sleeping mats, fruit baskets, but now it is being explored for ecological engineering purposes in the imizi project that I just talked about. So these increased demands of this plant could risk it going into overexploitation. And another issue with the plant is that the weaving culture of this plant is mostly held by elder generation, with the youth less interested. So the combination of these pressures could threaten the sustainability of the plant and also the practice of weaving. So for my chapter, I wanted to engage with the indigenous knowledge related to harvesting, cultivating, and weaving of imizi plant. Our objective was to document the cultural ceremonies and lived experiences associated with imizi and its traditional uses. We wanted to describe indigenous methods of cultivating, harvesting, and including the imizi availability. And we wanted to investigate the transmission of weaving knowledge across generations. So for this, I worked closely with two local communities in Eastern Cape, so the Amadiba and Hamburg. And the selection of these communities was based on their cultural ties with the plant. So the Amadiba is in Pondoland, in the Wild Coast. So they're largely relying on their gardens for food. They plant everything in their gardens. and they have strong cultural ties to the traditional weaving. And we also had Hamburg, which is also a rural community, coastal also in Ngushwa local community. Most residents, they rely on local shops for food. It is a well-established tradition of embroidery and handcrafts through the Keiskama Trust. And the imizi weaving is no longer being actively practiced. So we used this paradigm, which I struggle to pronounce. So it helps us to understand the meaning of people attached to their experiences, behaviors, and social contexts. And for the selection of participants, as I've said, we had Hamburg, and we had 30 adults from Hamburg, 28 females and 2 males. In Amadiba, we had 30 adults, and in Hamburg, we also had 10 youth, 6 females, and 4 males. So for data collection, we use semi-structured interviews, which are effective for capturing participants' lived experiences. So we selected an inductive approach where we used a questionnaire with open-ended questions. So there were 70 traditional knowledge holders, as I've mentioned. We obtained ethical clearance from Nelson Mandela University, and all ethical protocols were followed throughout the research, including informed consent in the languages that the participant understood, which is isiXhosa, and we also asked for permissions to record conversations and take photos. So the data was then translated and transcribed by me, fortunately, and I used a thematic analysis approach, which identifies and interprets patterns in the data. So I used a class TI for this, and it was an experience. So going to the results. So in terms of so the main structures that are made from this plant were the amakuko, or in singular ikuko. It is a sleeping mat, the name says it. So people use it for sleeping and mostly in cultural ceremonies. But before then, when there were no beds, we used the amakuko. And then we also saw izitebe, which is the flat mat used as a serving tray to serve food such as meat and boiled maize, inkobe. And there was also ineke, which is a collecting jar that is used for picking food items such as rice, flour, and maize. And so what we found is that imizi is central to almost every cultural ceremony used in umgidi, which is the celebration of when the young initiates come back home. Imbeleko, the introduction of a newborn, isipehlo, initiation for traditional healing, and in weddings. So, and people also sell the raw plant, and also they sell the items they make. In terms of ecology, both communities described imizi as being highly adaptable plant. It survives the sun, it survives shade, it prefers, it loves water, but it also can grow in drought. In terms of cultivation, Hamburg community does not cultivate, and they see cultivation as being offensive to the ancestors. So one participant even said that I can't bring the river to my home. In case of Amadiba, they still cultivate it, and they cultivate it in their gardens, and they have their ways of cultivating the plant, which were better than mine. In terms of harvesting, so in Hamburg, they do harvest, wild harvesting, but they have to ask for permission first from the ancestors before they harvest. And in Amadiba, they just harvest when they can, because their harvesting is associated with practicality and economy. And in terms of weaving, Hamburg, they do not longer practice weaving. They only practice weaving when the imizi project came to them. Some could weave, some had to learn during the project. And in Amadiba, they still practice weaving, but their issue is that their youth is no longer engaged. So from this, we learned two complementary models of sustainability, that Amadiba has adaptive cultivation system, which emphasizes ecological timing and active management, and Hamburg approach is rooted in sacred respect and minimal disturbance. So together they show indigenous ecological knowledge and cultural values. They safeguard biodiversity, foster resilience, and provide sustainable ways of living among environmental change. We also saw that there was erosion of knowledge, where the youth is no longer practicing this. And in terms of biocultural rights, so the new projects, the new ecological engineering projects that are coming through. So there will be a shift in governance and ownership issues related to indigenous plans. So early engagement with the right holders and respect for community protocols are essential. In conclusion, I will say ecology, culture, livelihoods are inseparable. Thank you.
Thank you so much, Will, for a lovely presentation. Okay, that's fine. Any questions from the floor?
Well, thank you very much for your presentation. My question is just simple. With the results that you got from your study, are you planning to go back to the communities just to educate the public about what you got from the results, and for conservation also aspect, just to engage the community about what you got on your project? Is it something that you're planning to do in the near future?
Okay, thanks, Kate. Actually, we have a planned event with Francesca to go back, because the other chapters were looking into similar things, but using science knowledge. So I was planting this and harvesting. So I will go back to give feedback and also show them what the other communities are doing. So I will.
Thanks for the great talk. So with socioeconomic aspect becoming more common in the science field, what are the challenges, some of the challenges that you experience while doing this project, and any advice for people that want to incorporate the people into science?
Can I ask if you're meaning socio-economic or socio-ecological? Okay. Okay, for me, it was a different space. I had to learn a lot, but it's doable and it's nicer. It fills more gaps than the science that we do. So I think it's worth getting into that space.
More questions? Okay. Because I'm a plant person, and you were talking about a plant here. So I'm just curious. I can't help it. Do you know any biotic factors like plant diseases? You know that this plant is susceptible to.
Okay, this one actually is
Considering its importance, yeah.
It is resilient, Kakul.
Oh, yeah.
So it came up in Dabaye, the worms. So they do eat it, but they are not, it can survive that.
Oh, okay.
Yeah, so no diseases that are recorded from the plant.
That's good.
Yeah.
That's nice. Right, thank you so much. A round of applause for Vuyo. I love the questions. Keep them coming. We need more. We almost at the end of our presentation. We have like 3 speakers, and then we're done. Our next. Okay. Our next speaker is Sipelele. He will talk about natural versus artificial coastal surface complexes, insights from in situ 3D imaging. Over to you, Ismail.
All right, good afternoon to everyone. Let's jump straight into it. So coastal environments are productive systems shaped by biotic and abiotic factors, with structural complexity being one of the key factors that influence biodiversity. Now, structural complexity describes the three-dimensional physical structure of the habitats. High complexity creates a variety of microhabitats, nursery, refuge, and spawning, which is critical for early life stages of marine invertebrates. In addition to this, there are biogenic species such as your mussels, barnacles, and invertebrates, mussels, barnacles, and oysters, which create what we call biogenic habitats, and that further enhances complexity. However, coastal urbanization is increasing, meaning there's a further expansion of coastal infrastructure, and that ultimately means that natural habitats are being replaced by artificial habitats. The structural complexity of natural habitats is intrinsically different from artificial habitats just by using this example visually. Therefore, the aim of this chapter was to compare and contrast natural and urbanized intertidal systems' structural qualities by assessing one, surface complexities, and two, the differences in those complexities between a biogenic rock and bare substrate. This was done across 6 study sites, the small ports, which is Port St. Francis and Port Alfred, the natural sites, which is Kinney Bay and Cannon Rocks, and then the large harbors, Port of Mucha and Port of PE. This was done by using the Xbox Connect V2 scanner, where I scanned a minimum of 6 3D scan models per habitat substratum per site, and I looked at 2 metrics, surface area and rugosity. Now, for the approach under both of those matrices, we're going to look at bare rock and biogenic rock. And then further, for the rugosity, we added multiple scales, which is your 5, 10, and 50 millimeter across all the 6 study sites. These are the results. What you need to know are 2 things. One, the ticks represent sites that are within the same postdoc grouping. 2, the red circles represent a site that has the highest surface area or rugosity. So in essence, looking at the surface area first, for the bare rock, the small marinas and ports had higher surface area than the other sites, and this was mainly due to the large rocks and the arrangement in how the sites were constructed. 2, the biogenic rock, the natural sites equal to urban sites, as you can see with the non-significant p-value there. And we suspect that the presence of mussels makes structural complexity similar between natural and urban habitats. And then for the rugosity, thank you. And then for the rugosity, both bare rock and biogenic rock produced a similar result in that at the 5 and 10 millimeter scales, the natural site had more rugosity than urban sites. However, at the 50 millimeter scale, urban site equal to natural site. Although I've ticked Kinney Bay there on the far right, if you see the other tick at the bottom, that's an urban site. So what does this all mean? Natural habitats have increased fine scale habitat heterogeneity at the 5 and 10 millimeter scales, while at the 50, natural habitats equals to urban habitats at the core scale of 50 millimeters. In addition to that, urban habitats have increased surface area. However, increased surface area alone does not translate into usable space. So these 2 differences between the natural and the urban, they overall influence the early stage processes such as your recruitment and settlement, and then the composition of populations of marine invertebrate species. And then the take home for this is that variety of complexity traits need to be taken into account during development of coastal infrastructure. Thank you.
Thank you so much, Ismail. Any questions? One question from the floor.
All right, thank you. Is that sensor waterproof?
No, it's not. So the sampling was done during low tide. And so I gave myself a period of about an hour, let's say max 2 hours before the low tide, and then 2 hours after, just to get enough time to scan, all the surface areas that I was interested in.
The round of applause was good. Thank you so much. Our next speaker is Katlego. He will talk to us about the potential of mussel beds as blue carbon habitats for climate change mitigation. Over to you, sir.
Good afternoon, everyone. My name is Katlego, a PhD student under the supervision of Francesca and the following core supervisors. So my project focuses on the mussel beds as blue carbon species. So carbon dioxide is known as the major contributor to greenhouse gases leading to global warming as well as climate change. While on the other hand, we know that marine environments are known to be major carbon sinkers through mainly through vegetated habitats such as salt marshes, mangrove, as well as sea grass, which these species in fact have established themselves through research work as blue carbon species. So now to supplement now the blue carbon ecosystems, mussels are identified for their potential to sequester carbon in tissues, shells, as well as in basses. But however, this information within our region has not been, in fact, no studies, in fact, have been presented, in fact, or on blue carbon habitat, on mussels, in fact, as blue carbon species. So now this is where now my research comes in. So the aim of my research is to investigate carbon uptake in two intertidal mussel species, Methylas galloprovincialis and Penna penna on the South African coastal regions, focusing on both natural and urban sites. So I have sites from the Eastern Cape as well as sites from KZN. That's where the sampling will take place. So now the first chapter of my project is to estimate carbon content in shells, tissues, and basal threads. So samples will be prepared by means of dissection, oven drying, homogenizing, as well as weighing of the samples. And carbon will be estimated by the usage of elemental analyzer. The second aspect of the project will look at the isotope analysis from mussel diet as well as tissues. This will involve sample preparations, and then this will be analyzed using the isotope ratio mass spectrometer. So now, carbon also will be estimated from respiratory measurements. So samples will be collected from the field and acclimatized according to their natural conditions. And thermal threshold will be estimated for both species, for both CT min and CT max. And a parasite system will be used to estimate the oxygen consumption for both species. And then we need to also calculate and estimate carbon released by means of respiratory measurements. So the last aspect of a project will be to estimate the mussel percentage cover on the South African coastal region. So this will involve the usage of drone sampling, images, satellite images, as well as quadrant sampling. So once we have all these images, we'll be able to estimate the amount of the abundance, in fact, of both mussels. And this will enable us in terms of quantifying the amount of carbon which is captured on mussel beds. So now, at the moment, I'm still looking at the pilot study, which seeks to optimize and standardize the methodology for both Chapter 1 and Chapter 2. So different size classes of the animal were collected from Canton and Sea, and then sample preparation. I'm still at a point of preparing my samples for carbon analysis. So now, in conclusion, the aim of my study, in fact, is to provide an equation that will represent a carbon cycle from mussel beds. This will enable us to integrate mussels within the blue carbon ecosystem. Thank you very much.
So much, Kaseo. Questions from the floor?
Thanks, Kat. I don't know, maybe I don't know this, but I'm familiar with city mean and city max for fish, where you estimate it when the fish loses equilibrium. So how do you do that for mussels?
The estimation of what?
City man and city mix, city mean and city max.
Okay, we'll use a water bath system that will enable us to establish city max and city mean by means of ramping the temperature and as well as lowering the temperature of the animal. And then we'll look at the behavioral changes, the physiology, in fact, of the animal that will tell us that it has reached its thermal threshold or as well as the mean temperature. So it's just by using the water bath system and the system that ramps temperature up and down.
Thanks, Katlego. I think this is a very nice project with so much potential. But I'm just curious whether you've done maybe this sort of calculation already, where you look at the amount of carbon dioxide that's being produced and how much mussel beds would require to be able to have a significant maybe contribution in reducing that carbon dioxide that's being produced. How much in terms of, maybe just out of interest. Thank you.
Well, they don't capture as much as salt meshes like the vegetated habitats, but they do present themselves as in animals that can actually capture carbon, even store carbon within shells. And once the animal dies, in fact, that carbon is stored, in fact, within the shell, even on the ground or on the sediment, in fact. And that goes, in fact, it goes even up to the sediment itself of the mussel bed. But relative to salt, to vegetated habitats, I wouldn't say that they contribute much, but they are a potential in terms of capturing carbon within their tissues. Yeah. So it's just a
round of applause for Katlego. Ladies and gentlemen, this is our last presentation for today, and yeah, it is also a speed talk. I don't want to put your name. May I please call you Amir? That's good. Okay. He will talk to us about assessing the blue carbon storage in coastal ecosystems.
Hello, everyone. I'm Amir Hassan Masumi from Iran, and my PhD thesis is on the potential role of mussel beds in blue carbon. What is blue carbon? The carbon is captured and stored in marine and coastal habitats is blue carbon. Why these habitats are important? Because they are carbon sequestrers, which play a significant role in climate change mitigation. Although there are lots of studies on blue carbon, most of them consider vegetated habitats like mangroves, salt marshes, and seagrasses. We want to focus on mussel beds. My thesis is divided in 4 chapters. We want to evaluate carbon in mussel biodeposits. Then we'll go for decomposition. We want to assess carbon changes during decomposition of mussel shells. By these 2 main steps, we'll go for determining carbon in we go for determine mussel-derived carbon in adjacent coastal sediments. And the final chapter is comparing carbon storage in receded mussels in different habitats. For the first chapter, we want to evaluate carbon content in mussel biodeposits in two species of Mytilus galloprovincialis and Perna perna in different treatments. Our hypotheses are, first, small specimens will produce more biodeposits than large ones by their weight due to higher rate of metabolism. The next one is mussels will produce more biodeposits in single tanks than group tanks due to less competition. And the last hypothesis is Mytilus galloprovincialis as an invasive species will produce more biodeposit than Perna perna, which is an indigenous species. After mussel collections, we will keep them under laboratory condition, and after a certain time, for example, 24 hours, we'll collect biodeposits by syringe and prepare them for carbon analysis by elemental analyzer. The next step is decomposition of mussel shells in both situ and in situ and ex situ habitats. Although ties and erosions in natural habitat batter mussel shells, revealing the time of decomposition in laboratory experiment enable us to assess not only the duration of decomposition, but also the fate of carbon. We will utilize both broken and fine shells to evaluate decomposition in laboratory. The next chapter is collecting sediment samples by cores from mussel beds and as well as different distances from mussel beds to determine the amount of carbon in those sediments by elemental analyzer. And another point is that although there are lots of methods for carbon provenance, most of them show overlap between different carbon sources. So we will use metabarcoding. And the last chapter is fixing different population of mussels in different habitats by pipe and eyebolts. And after a certain time, for example, every 3 months, we will extract some of them and evaluate them for carbon content. After completing this project, we will fill the gap of mussel beds in blue carbon studies, publish 4 papers, attending in 1 national and 1 international conference. Thanks for your attention.
Thank you so much, Abdul. Any questions from the floor?
Thank you for the talk. I study carbon also, so I'm excited to hear about carbon. But because of my limited knowledge on what you are trying to do, I'm trying to understand, yes, you would look at the fate, but what do we know in terms of, in literature, the resident time of this carbon in these mussels?
In which chapter you mean? You mean in sediment, you mean, or in the decomposition?
The overall, for it to contribute long-term sequestration, we need to know the resident time. That's what I'm interested in.
When we go for collecting cores, sediment cores, we can identify the amount of carbon there. So by comparing that amount with the quantifying the DNA for mussels in those sediments, we can easily shed the light on that. These carbon came from mussel beds. And by another thing is that when we will collect sediments in different distances from open water. So by this, we can compare that those older sediments on high shores with new sediments in low shores. I hope I can answer your question.
Thank you for that. So I'm glad my question from the previous speaker wasn't asked, because I think your talk covers everything I wanted to talk about. For your sediment chapter, where you're going to trap what you were just talking about, I wonder if the fact that you're going to use a sandy beach is going to affect the outcome, because we know with our bigger particle sizes, there's more flushing. So I wonder if that's going to influence your outcome. And I'm wondering if that experiment shouldn't be done adjacent to an estuarine mud bank, for example, where your muddy sediments create an anoxic situation that's going to, which is why, you know, mangroves and salt marshes have that carbon stock, because it's anoxic, whereas your sandy beaches may be more oxic and have more flushing. So just something to think about. Maybe it's not a question, maybe it's just for you to note. Yeah. Thanks.
Okay, I think we can have one more question and then we'll.
It has been found out that mussels, in fact, shells can capture carbon for a long extended period of time. Even when the animal dies, you still have those shells, in fact, remaining on the mussel bed. So in terms of long-term storage, we can bet on mussels, in fact, also as well.
Thank you very much. Thank you for a lovely talk. I just want to give a round of applause to all of the speakers. You have been absolutely awesome. I think this is the very, very first symposium I've ever been to in my entire life that everyone's kept so well to time. So well done, everybody. You guys have done brilliantly. So give a round of applause to that. Well done. Just to close off, I'm going to ask Julie if she wouldn't mind giving some closing remarks. And then after Julie's spoken, we'll give the awards for Best Speaker, Best Speed Talk, and Best Poster.
Ignore me. It's nice and cool down here. I think that the heat is still high at the top. So thanks everyone. Thanks to our students. Thanks to your supervisors. Your highly intelligent supervisors. And of course, the organizers for making the symposium such a success. To the organizing team, Gwyneth, Jody, Etienne. I don't know the rest of the team. There we go. Thank you so much for putting together an excellent program, a well-run symposium, excellent catering, and of course, very good timing. I think let's give them a round of hand. It's no mean feat to get us all together in both in person and virtually. I don't know if anyone is still on virtually, but to celebrate the culmination of another year, or maybe 2 or maybe 3 years worth of research. What we've seen today is more than just research, though. It's a powerful demonstration of what our national research tongue-tied. It's the heat. Infrastructure platform like SAIAB enables. Without this, I doubt that many of you would be where you are today. We've heard about studies using cutting-edge genomics,
using AI and information system technologies, ecophysiology techniques, and all of these across freshwater ecosystems, estuarine ecosystems, marine ecosystems, as well as deep sea exploration using things like ROVs and I don't know what they're called, but fancy machines that go ping. Taryn, that's your acoustic telemetry things, I'm assuming. And all of your work really reflects the full spectrum of our Institute's capabilities. So thank you for using those platforms. Thank you for using the research infrastructure that the NRF provides to you all. The ships and the boats and the ROVs that allow us to collect this biodiversity data, the labs that unlock these molecular insights, Jodie and Gwyneth, and the collaborative networks that connect us are not just resources for you all, they're also catalysts for discovery. And I think today's presentations really show how these tools empower you guys to ask very big, bold, important questions. And I think most importantly is to deliver answers that matter. You know, we're not accountants or lawyers are important. But, you know, we're delivering answers for science, for the environment, and it's so great to see answers for society. The socio-ecological studies that are starting to pop up or have always been there. I mean, I'm new to SAIAB, new-ish. It's really a focus that we have to, or a field that we need to incorporate, because I don't know if any of you went to, where's Prof. Potts? Anyway, without environment, we can't have functioning societies. And without societies, we can't have functioning environments. So that's really important as we go forward. So I want to encourage you guys to keep pushing boundaries, keep using this infrastructure that we provide to tackle these big challenges. And as we speed downhill towards the end of the year, you guys might be frantically analyzing data. If you're submitting, I hope you're not still collecting data. I'm sure you're writing up your data. I just want to ask you all to take a moment, or moments, many moments, to be kind to yourselves. Appreciate how hard you've worked and still have to work. Appreciate how far you've come and how much further you're still going to go. Also, please be kind to those around you. Be kind to your families who support you, your friends who support you. I'm sure there's many late nights and tears, maybe, tantrums, binges. But be kind to those people around you who are supporting you. And also to your colleagues and supervisors who might be helping you to get to your submission day. I also want to ask you to be kind to those in the background, those who make your daily lives easier. Our admin staff, our technical staff, our cleaning staff who are always there making the life of us at SAIAB a whole lot easier. And finally, guys, be kind to strangers online. I have posted some content recently on the CBC's Facebook page, and people are nasty, vicious, unkind, judgy. Just be kind. I don't know if anyone watched Ted Lasso, but he sat back once and said, be inquisitive, not judgmental. And take that home with you. Today, we recognize the completely inadequate efforts of our country to curb GBV. And so a smile, a nod, a subtle acknowledgement might go a very long way in helping someone who is struggling in that space to have the courage to perhaps stand up against an abuser. And so I want to, I don't know, is this a call to action? Let's make kindness part of our research culture. Let's create spaces where respect and empathy are as fundamental to our daily lives as scientific rigor and innovation are. And with that, I'd like to thank you all. I hope you celebrate this evening or study. Maybe you've got some stats, some R code to work on. And if you're traveling, travel safely. And yeah, here's to an excellent 2025. Here's to belonging to an excellent institution. And congratulations, everybody.
Great. So last things before we carry on. First of all, I'd like to thank both of our keynote speakers. Unfortunately, Professor Dorrington couldn't stay with us. She had another engagement. But thanks to Professor Dorrington and Professor Rajakan, thank you so much for being part of our symposium and for sharing their wealth of knowledge and experience with us. So I've got a small thank you gift. Anusha, would you mind coming up? Okay, so we're just adding up the final scores for the talks, etc. While that's happening, I just want to ask you all, please, your name tags, leave them on the table outside. For those that left a little earlier, grab your friends and tell them to give me the name tags back so we can reuse them next year. Waste not, want not. So please hand them back, collect them from your friends who are not here anymore, pop them at my office or my pigeonhole. You can keep the inside bits, the piece of paper if you really want to, but name tags. For those of you that had posters, you feel free to take your posters home. They are your posters now. If you want to stick them up on your office wall or in the passageways, you're more than welcome to. And the last thing I want to mention is this is a new type of format for the symposium. I will welcome any and all feedback, what worked, what didn't work, what you would like to see in future symposia, and what you would like to keep for future symposia. So thank you. And most of all, thank you to my wonderful team. I have got the best team ever. So thank you guys for all your hard work. Couldn't have done it without you. So with Okay, so we're going to start off with the posters. Michonne Duplessis won the best poster. Is she around? Oh. Tell Michonne to come find me. Okay. Michonne Duplessis. For the full length of the standard talks, it was a tie. Okay, one between Luther Adams and Buhle. Well done, guys.
Let me stand for first. Let's stand.
Take lots of photos of Jody still adding up for the last bit for the speed talks. Almost there. No pressure. No pressure. Poor old Jodie's sweating bullets here. Body and then punch.